Hi class, today we learn on the last chapter which is cells reproduction including cell cycle and also protein synthesis. But this week we will only learn on three subtopics which are genes and chromosomes, mitosis and also meiosis. So what is the importance of cell division? So the ability of organisms to produce more of their own kind is the best way to distinguish between living things from non-living things. So the continuity of life is based on the reproductions of the cell, or also known as a cell division. The divisions of unicellular organism will reproduce the entire organism of the unicellular, increasing the population. Okay, and also cell division enables sexually reproducing organs to develop, such as a multicellular organism from single cells to the fertilized egg or the zygote. So division also important for the renewal and repair by replacing the cells that die from the normal wear and tear or accidents. So, as I mentioned earlier, unicellular organisms, the divisions of one cell reproduce an entire organism, such as an amoeba. However, for multicellular organism, the division of cell does not reproduce the entire organism, and it depends on cell divisions for the development from for growth and development from a fertilized cells, or for the repair and the tissue renewal, such as the dividing bone marrow cell that will give rise to the new blood cells. So let's look what is inside the cell. So inside cell there is a nucleus that control all the activities inside the cell and contain a genetic material. Genetic material here is referred to the DNA, in which the DNA is entangled and wrapped or coiled and forming the chromosome. Okay? So all the DNA in a cell constitute the cell genome. So all of the DNA that you can find in organism is known as the genome. So in prokaryotes, the genomes, okay, is often a single long DNA molecule. Meanwhile, in eukaryotes, the genome consists of a number of DNA molecule, much more complex compared to prokaryotes organism. So, such as the bacteria is one of the example of the prokaryotes, in which they reproduce by a type of cell division that known as a binary fission, in which the plasma membrane here, they will pinch inward, they will go inward, thus dividing the cell into two. For eukaryotes, okay, cell, mostly multicellular cells, the chromosome consists of chromatin, okay, that chromatin is actually a complex of DNA, or also known as a chromatin, also known as a chromosome, and the protein that condenses during the cell division. So let's look further on the eukaryotic chromosome, either what inside the eukaryotic chromosome and the structure. So eukaryotic chromosome consists of chromatin, a complex DNA, and protein that will condense during the cell division. DNA molecule are packaged into a chromosome. This is the DNA molecule. They will wrap the protein histone and then package into chromosome. So the nuclei of human somatic cell is referred to non-reproductive cell. They have two sets of 23, meaning to say 23 chromosome multiplied by 2 sets. So somatic cell contain 46 chromosomes. Meanwhile, the reproductive cell, or known as the human gamete, such as the sperm and also the eggs, they have half set of the 23 chromosome. That means they have 23 chromosomes. So let's look further on the chromosome structure. Chromosome structure, one of these is known as the chromatid. So here we have two identical chromatid in which one is exact copy of the other, and each of these two identical chromatid, they contain one DNA molecule. We call it also as a sister chromatid. And this part here is known as a P arm because it is a very short arm structure. Meanwhile, at the bottom here, the bottom part here, the length is much more long compared to the upper part, and this part is known as the Q arm or long arm structure. And chromosome also has a centromere in the middle, okay, here between the P arm and also Q arm, in which this is referred to the constricting point of the chromosome where the two chromatid will touch each other. And next, of course, inside the chromosome there is a DNA molecule that being coiled and folded, forming the chromosome structure. So the chromosome, inside the chromosome there is a chromatin, and chromatin is actually made up from the nucleosome, and nucleosome is actually referred to the DNA molecules that wrap the histone protein. And there are two types of cell division. We have mitosis and meiosis. Mitosis is referred to the divisions of cells that produce two daughter cells which genetically identical to the mother cells. Meanwhile, for meiosis, it is referred to divisions of cells that produce four daughter cells, and the genetic content is different or varied compared with the mother cell. And next, for mitosis is under the cell cycle, in which cell cycle is actually an ordered sequence of events of in life of the cells from its origin in the divisions of the parent cells until its own divisions into two. So the mitotic phase here is alternate with the interphase in the cycle. So first we have the mitotic phase, and then next we're gonna have the interphase, in which including the G1, S, and also G2 phase. So the cell cycle, of course, as I mentioned earlier, consists of interphase and also mitotic phase, in which my interphase is important for the cell growth and also copying the chromosome in preparation for cell division. Meanwhile, mitotic phase or mitosis, including the mitosis and cytokinesis, that one is for cell division. So interphase here, okay, can be divided into another three subphases, which is G1 phase, S phase or known as a synthesis phase, and G2 phase or known as a second gap phase. So the cell will grow during these three phases. However, the chromosomes are only duplicated during the S phase, which is the synthesis phase of the cell cycle. So now we look on the mitotic phase of the cell cycle, which is involving mitosis and also cytokinesis. So as I mentioned earlier, mitosis is a cell division that produces two daughter cells, and in which the daughter cells are genetically identical to the mother cells. So the chromosome of the daughter cell, of course, have the same number of chromosomes with the mother cell. And mitosis is very important to increase the number of cells for growth, to repair and replace the damaged or dead cell, to produce daughter cell which genetically identical to mother cells, and it is the basis for asexual reproductions of unicellular organism. That's why the offspring is identical to the mother cell. So this is the overview of the mitosis, in which the first one, what will happen, the eukaryotic cell, they has a multiple chromosome. However, figure here only show one chromosome. And before duplication, each of these chromosome, there has a single DNA molecule inside this chromosome. Next, what happened? DNA duplication will take place, in which then it will result the two sister chromatid that will connected at the centromere. Okay, so each of these chromatid, they contain the DNA, the same DNA molecule. Next is that mechanical process will separate these two sister chromatids into two new chromosomes, and then they distribute them to become two daughter cells. So mitosis consists of four main stages, including prophase, metaphase, and telophase. And after mitosis, we do have cytokinesis process that involving the cell plate formation in plant cell and cleavage furrow formation in animal cells. So before we look on the process that involve in mitosis, let's know the important terms in cell division. First, we have mitotic spindle. Mitotic spindle is a structure that made of microtubule, okay, that will control the chromosome moving during the mitosis. Okay, for centrosome is the microtubule organizing center, assembly of spindle microtubule that has two chromosomes, a centriole. Meanwhile, Aster is actually a radial array of short microtubule that extend from each centrosome here. Meanwhile, the centromere, as I mentioned earlier, it is the specialized regions of the chromosome where the two sister chromatids are most closely attached. And last but not least, we have a kinetochore. Kinetochore is a structure of proteins that attach to the centromere that links each sister chromatid to the spindle fiber. So this is Aster that produced from the centrosome. Centrosome here consists of one pair of the centriole, and then we do have a kinetochore, okay, the one that will attach to the spindle fiber, and this yellow in color is referred to the spindle fiber. So spindle fiber made of microtubule. So microtubules are hollow rods about 25 nanometer in diameter and about 200 nanometer to up to 25 micron long. And microtubule made up from a globular protein that called as the alpha tubulin and beta tubulin. So the two-dimensional sheets of the tubulin later will be rolled. Here, as you can see, they are rolled into a tube, and they can elongate by adding the microtubule units to one end here. Okay, and the mitotic spindle or microtubules, they also can disassemble or depolymerize or polymerize inside the cell. So overview of the mitosis after the G1-2 phase, and then it will undergo mitosis, including prophase, metaphase, anaphase, and telophase. Next, it will end it with the cytokinesis. Okay, so what is interphase? Interphase is a stage before the prophase of mitosis. So this is the preparation stage for mitosis process that consists of the first gap, synthesis, and also the second gap phase. So in the nucleus here, there is a tiny thread called as the chromatin, which start to replicate themselves during the S phase or synthesis phase. So what will happen during the S phase? The chromosome will duplicate. However, they cannot be seen individually because they have not yet condensed. Okay. So the two centrosome will happen here and form by duplicating a single chromosome centrosome, and each of these centrosomes contain two centrioles, meaning centrosome is made up from two pair of centrioles. So four centrioles in total. Next is mitosis. The first stage of mitosis is prophase. So during prophase, chromatin slowly condense into chromosome. As you can see here, it start to forming the chromosome, where they will become shortened and very thick. And each of these chromosome consists of two chromatid, one and two, that attach at the centromere. Then the nuclear envelope here, they will start to disintegrate and also disappear, and the mitotic spindle or the microtubule will grow out from the pair of this centrosome and migrate. Each of them will migrate to the opposite poles of the cells. This first of the centrosome will go here. Meanwhile, the other centrosome will move to the other side. And then during the late prophase, the spindle microtubule will attach to the kinetochore of the chromosome. So, before metaphase process, there is another process that called as the prometaphase. So, during the prometaphase, the spindle microtubule here they will interact with the chromosome. Okay, so each of the two sister chromatid of each chromosome has a kinetochore. This is a kinetochore. Kinetochore is referred to the part of the chromatid that will attach to the spindle fiber. And some of the kinetochore, the microtubule will attach to the kinetochore. This one is known as a kinetochore microtubule. And non-kinetochore microtubule interact those from the opposite pole of the spindle. This one, the spindle fiber that does not attach to the kinetochore, known as a non-kinetochore microtubule. So next, all of the chromosome will move toward the metaphase plate. So what is metaphase? Metaphase is the stage where the chromosome will line up at the metaphase plate. Okay, so for each chromosome, the kinetochore here of the sister chromatid are attached to kinetochore microtubule. This one is non-kinetochore microtubule because they do not attach to any chromosome. And for the kinetochore microtubule, they will pull the sister chromatid to the opposite pole. And during anaphase, the anaphase is the shortest stage of mitosis, which lasting for few minutes. So the attachment between two sister chromatid of each chromosome, they will break here. As you can see, they separated from each other, and these two sister chromatid will move to the opposite poles of the cells, and the movement of chromatid is due to the shorten of the mitotic spindle or kinetochore microtubule. So let's look on how does the kinetochore microtubule have been shortened. So let's look here. During the experiment, this is the sister chromatid, single chromatid that attached to the kinetochore microtubule. What will happen? We will mark over here at the kinetochore microtubules, and then during the process, during the experiment, what will happen? The chromatid become very close to the line that have been marked. Therefore, this part of kinetochore microtubule has become shortened. Okay, they become shortened due to the depolarizing of at the kinetoco end. As you can see here, this is the chromatids, okay, the chromosome. When they move toward the pole, what will happen? They will polymerize the microtubule to become the tubulin subunit. Next is telophase, the last stage in mitosis before cytokinesis. So during telophase, both set of chromosomes will reach the opposite poles of the cells, okay, as you can see here. And the chromosome will become very less dense and become a thread of chromatin again. And the spindle microtubule is depolymerized. That's why you cannot find any microtubule here except for the Aster. And the nuclear envelope and the nucleolus will reappear again. Next is cytokinesis, the phase that will complete the mitosis process. Cytokinesis also refers to the cytoplasm division. If you look on the mitosis is referred to the nuclear divisions. Meanwhile, cytokinesis is referred to the cytoplasm division. So in animal cells, the cytokinesis occur by the process that known as the cleavage, in which they will form a cleavage furrow here that will pinch inward and separated the animal cell becoming two daughter cells. Meanwhile, in plant cell, okay, the cell plate will form during the cytokinesis. Okay, as you can see here, the cell plate is formed that that derive from the Golgi apparatus. So the vesicle that contain the cell wall materials and it will fuse together. Okay, a vesicle here that made up from the Golgi apparatus. They will fuse together and they will produce a cell wall. So this cell wall will be enlarged and later fused with the plasma membrane, and thus separating these two daughter cells. So this is how it look like. This is onion, not an iron. Okay, typo here. The prophase that happen in onions roots here, prometaphase and metaphase plate, where the chromosome arranged in the metaphase plate. During anaphase, each of the sister chromatid will move toward the opposite pole, and then during telophase, the cell plate will form, thus separating these two daughter cells. Okay, so in conclusion, mitotic phase including prophase, metaphase, anaphase, and telophase with cytokinesis. So, what is the differences between mitosis in animal and plant cell? So, in plant, there is no centriole and ester, but animal cell, they do have a centriole ester that come from the centrosome. And cytokinesis in plant involves the formation of cell plate. Meanwhile, cytokinesis in animal cell involves the formations of the cleavage furrow. And in plant cell, the division is occur in the meristemic cell. Meanwhile, in animal like human, they